Dear students, after studying this module, you shall be able to learn and understand the Hardy-Weinberg equilibrium, understand the factors affecting Hardy-Weinberg equilibrium, correlate the genotypic and allelic frequency distribution, comprehend how population evolve in nature. The basic principles of heredity that are the cornerstones of modern genetics were found by Gregor John Mendel while working with garden peas. However, the work of Mendel remained obscure for about 3 decades until its rediscovery in 20th century. However, controversy prevailed for several years as it was not then known how it could explain the continuous characteristics. In the year 1902, Udne Yule debated against Mendelism as his belief was that in a population the dominant would increase. In the year 1903, William E. Cassell revealed that the frequencies of genotype will remain stable even without selection taking place, and in the same year Karl Pearson propounded that a single equilibrium point having values wherein both p and q were equal to value of 0.5. Reginald Punnett introduced Euler's point as a problem to a British mathematician named G. H. Hardy. Thus, this principle came to be known as Hardy's law until Kurt Stern pointed out in the year 1943 that the principle had been independently postulated by the German physician Wilhelm Weinberg in the year 1908. While in the year 1903, William Cassell derived the ratios for the special case of frequencies with equal and hence is rarely known as Hardy-Weinberg-Cassell law. The Hardy-Weinberg principle is one of the simplest and most important principles in population genetics. To illustrate the principle, consider a large population of sexually reproducing organisms. The organisms are assumed to be diploids, meaning that they contain 2 copies of each chromosome, one received from each parent. The gametes they produce are haploid, meaning that they contain only one of each chromosome pair and are in accordance with Mendel's law of segregation. During sexual fusion, 2 haploid gametes fuse to form a diploid zygote, which then grows and develops into an adult organism. Almost all multicellular animals, with exceptions such as drones of a bee colony, etc., and many plants have a similarly patterned life cycle. Consider that a given locus in an autosome, there are 2 possible alleles, dominant A and recessive A. Since each gamete can only have either of the 2 alleles, dominant A and recessive A, there are 3 possible genotypes in the population: homozygous dominant A, heterozygous AA, and homozygous recessive AA. Organisms with homozygous dominant A and homozygous recessive A genotypes are called homozygotes. Those with heterozygous AA genotypes are heterozygotes. The proportions or relative frequencies of the 3 genotypes in the overall population may be denoted FAA, F heterozygous AA and F homozygous recessive AA respectively, where F homozygous dominant A plus F heterozygous plus F homozygous recessive is equal to 1. Assuming that the genotypic frequencies are the same for both males and females, the relative frequencies of dominant A and recessive A alleles in the population may be denoted P and Q, where P plus Q is equal to 1. The Hardy-Weinberg principle gives the relationship between the allelic and the genotypic frequencies. It states that if mating is random in the population and if the evolutionary forces of natural selection, mutation, migration and drift are absent, then in the offspring generation the genotypic and allelic frequencies will be related by the following equation: F homozygous dominant A is equal to P square, F heterozygote AA is equal to 2PQ and F homozygous recessive is equal to Q square. For a population to be at Hardy-Weinberg equilibrium, 5 parameters are necessitated: no immigration or immigration, no change because of mutation in the allele frequencies, random mating, a large breeding population, no natural selection. It is noteworthy to mention such populations in nature are extremely rare, if at all present. A population where all individuals are potential partners and there are no mating restrictions based on either genetic or behavioral components upon the population, and that therefore all recombination is possible. Such a population is called panmictic population. A large breeding population ensures that chance alone does not disrupt genetic equilibrium, meaning that genotype and allelic frequency doesn't change over the generations. Consider a large population of n individuals of a diploid sexually reproducing species with an assumed sex ratio of 1:1 with genetic composition at some locus A at the breeding adult stage of a given generation F1. Such a population can also be described in terms of an array of allele frequencies, which in turn are related to be genotypic frequencies. These breeding adults will produce gametes that can all be presumably collected into a large gametic pool. Supposedly, the fertility is not associated with locus A genotype and that mutations are not taking place in cells that will result in the formation of gametes. It follows from that the frequencies of allele will not change in the transition from breeding adults to gametic pool. It's so because of our assumption that when we move from breeding adults to the gametic pool, the alleles that have been paired to form diploid individuals separate out and are copied to form haploid gametes, and also the number of copies formed are same as that of the number made for different alleles. Further, if all these gametes in the gametic pool fuse in pairs at random to give rise to a pool, then all possible diploid zygotes, a subset of which will represent the actual zygotes, will become the next generation. Millions of gametes are typically produced by each individual during its reproductive phase, and assuming that all of them fuse to form zygotes, we now have millions of zygotes in our zygotic pool. However, for a finite population such as ours, consisting of n individuals, the population tends to maintain constant numbers from one generation to the next. Once again, the statement actually contains two assumptions, namely that the number of breeding adults n is constant, and secondly, generations are discrete such that offspring do not coexist with their parents. Now we need to conceptualize how we get n actual zygotes that form generations F2 from millions of zygotes in the pool of all possible zygotes. Let us suppose that from the large pool we are doing the sampling of n zygotes and that it is purely random process with regard to locus A genotype. As we are doing the sampling on a very large pool, hence this sampling can be thought for all practical purposes to be with replacement. Also, it cannot be expected that the frequencies of the genotype changes as we move from pool consisting of possible zygotes to the pool of actual zygotes in F2 generation. In contrast, if a small population is considered, only a few copies of a certain allele may exist. The frequency of allele is bound to change in case the organisms do not reproduce successfully. In case of bottleneck event and genetic drift, non-selective random changes appear. Random mating. Random mating means the absence of a genotypic correlation between mating partners, that is, the probability that a given organism mates with homozygous A partner, for example, does not depend on the organism's own genotype, and similarly for the probability of mating with a partner of one of the other two types, heterozygote A or homozygous recessive A. Random mating will lead the genotypes to be the Hardy-Weinberg proportion, which is a consequence of Mendel's law of segregation. Random mating is in effect implies that offsprings are being formed by randomly picking pairs of gametes from a large gametic pool and fusing them into a zygote. The gamete pool contains all the successful gametes of the parent organisms. Assuming that forces of selection are absent, all parents must contribute equal numbers of gametes to the pool. According to the law of segregation, an heterozygous AA produces gametes bearing the A and A alleles in equal proportion. Therefore, the relative frequencies of the alleles dominant A and recessive A in the gametic pool will be the same as in the parental population, namely P and Q respectively. Given that the gamete pool is very large, when we pick pairs of gametes from the pool at random, we will get the ordered genotype pairs homozygous dominant A, heterozygous AA and homozygous recessive A in the proportions P square is to PQ is to QP is to Q square. With an organism, heterozygous AA and AA pairs can be regarded as equivalent. Above portions become P square is to 2PQ is to Q square, resulting in the Hardy-Weinberg proportions for the unordered offspring genotypes. Hardy-Weinberg principle can easily be extended to a trait governed by multiple alleles such as hair color in humans. For the multi-allelic traits, suppose there are n alleles at the locus A1 to AN with relative frequencies of P1 to Pn respectively, where P1 plus P2 plus 2Pn is equal to 1. Assuming that the population is large, random mating is taking place, evolutionary forces are absent and Mendel's law of segregation holds, then in the offspring generation, the frequency of A1 genotype will be P1 square and the frequency of unordered A1 genotype will be 2PQ. Irrespective of the initial genotypic proportions, random mating will automatically produce offspring in Hardy-Weinberg proportions. So, if generations are non-overlapping, that is, parents die immediately after reproduction, just one round of random mating can bring about Hardy-Weinberg proportion in the whole population. However, in case the generation overlap such as in human population, more than one round of random mating is needed. Once Hardy-Weinberg proportions have been achieved, they will be maintained in subsequent generations so long as the population continues to mate at random and is unaffected by evolutionary forces such as selection, mutation, etc. The population is then said to be in Hardy-Weinberg equilibrium, meaning that the genotypic proportions are constant from generation to generation. Population, genetic models of evolution and Hardy-Weinberg's principle. Natural selection. Charles Darwin's natural selection in terms of alterations in the gene pool implies that individuals having certain genes are better suited to produce mature offsprings than those lacking the genes. As a result, assuming that all individuals are equally fertile, the frequency of those genes will increase. Natural selection results from differentiated motility and or differential fecundity. Motility selection. Genetic makeup of certain individuals within a population allows them to be more successful than others in surviving through to the end of their reproductive period. The evolutionary impact of motility selection can be in effect any time following fertilization with the formation of a new zygote till the end of the fertile period of that organism. Motility selection is simply another way of describing Darwin's criteria of fitness, that is survival. Fecundity selection. Certain phenotypes may make a disproportionate contribution to the gene pool of the next generation by producing a larger number of young ones. Such fecundity selection is another way of explaining another criterion of fitness described by Darwin, that is family size. In each of these models of natural selection, certain phenotypes are better suitable than others in terms of their genotype to contribute their genes to the next generation and are thus more fit by Darwin's standards. The overall outcome of the two is a gradual change in the gene frequencies within that population leading to evolution. Population geneticists usually define evolution as any change in the genetic composition of population over a given period of time. The four factors that can bring about such a change are natural selection, mutation, random genetic drift and migration into or out of the population. In addition, changes to the mating pattern can alter the genotype without affecting allele frequencies. Selection at one locus. Natural selection occurs when some genotypic variants in a population enjoy a survival or reproduction advantage over others. The simplest population genetic model of natural selection can be assumed to comprise of an autosomal locus with two alleles, dominant A and recessive A. The three resultant diploid genotypes, homozygous dominant A, heterozygous AA and homozygous recessive A, have different fitnesses denoted by W11, W12 and W22 respectively, that are assumed to be constant across generations. A genotype's fitness may be defined in this context as the average number of successful gametes that an organism of that genotype contributes to the next generation, which depends on how well the organism survives, how many matings it achieves and how fertile it is. In case one of the above genotype is favored by natural selection, then genetic composition of the population will change. Consider that prior to operation of selection forces, the zygote genotypes are in Hardy-Weinberg proportions and the frequencies of dominant A and recessive A alleles are P and Q respectively, where P plus Q is equal to 1. The zygotes then grow to adulthood and reproduce, giving rise to a new generation of offspring zygotes. Our task is to compute the frequencies of dominant A and recessive A in the second generation. Let us denote these by P dash and Q dash respectively, where P dash plus Q dash is equal to 1. In the first generation, the genotypic frequencies at the zygotic stage are P square, 2pq and Q square for dominant A, heterozygous AA and homozygous recessive A respectively by the Hardy-Weinberg law. 3 genotypes produce successful gametes in the proportion to their fitness, that is in the ratio W11 is to W12 is to W22. The average fitness in the population is W is equal to P square W11 plus 2p Q W12 plus Q square W22, so that the total number of successful gametes produced is NW, where N is population size. Assuming that no mutation occurs and that Mendel's law of segregation holds, then an homozygous dominant A organism will produce only A gametes, and homozygous recessive organism will produce only A gametes, and an heterozygote AA organism will produce dominant A and recessive A gametes in equal proportion. Therefore, the proportion of dominant A gametes and thus the frequency of dominant A allele in the second generation at the zygotic stage is P dash is equal to n P square W11 plus half n 2pq W12 divided by NW is equal to P square W11 plus PQ W12 by W. Equation 1 is known as recurrence equation. It expresses the frequency of dominant A allele in the second generation in terms of its frequency in the first generation. The change in frequency between generations can then be written as P is equal to P dash minus P, which is equal to P square W11 plus PQ W12 by W minus P is equal to PQ by P into W11 W12 plus Q W12 minus W22 by W. If P is greater than 0, then natural selection favored A allele to increase in frequency. If P is less than 0, then selection has led recessive A allele to increase in frequency. If P is equal to 0, then no change in gene frequency has taken place and the system is in allelic equilibrium. However, the condition P is equal to 0 does not imply that no natural selection has occurred. The condition for that is W11 is equal to W12 is equal to W22. It is possible that gene frequencies have remained unaffected despite the natural selection. Equations 1 and 2 precisely indicate how fitness differences between genotypes will lead to evolutionary change. This enables us to explore the consequences of various different selective regimes. Suppose firstly that W11 is greater than W12 is greater than W22, that is the AA homozygote is fitter than AA heterozygote, which in turn is fitter than AA homozygote. By inspection of equation 2, we can see that P must be positive. So, with each generation, the frequency of dominant A allele will be greater than in the previous generation until it eventually reaches fixation and the recessive A allele is eliminated from the population. Once the A allele reaches fixation, that is P is equal to 1 and Q is equal to 0, no further evolutionary change will occur. If P is equal to 1, then change in P is equal to 0. This makes good sense because the A allele confers a fitness advantage. Therefore, its relative frequency in the population will increase from generation to generation until it is fixed. It is obvious that analogous reasoning applies in the case where W22 is greater than W12 is greater than W11. Equation 2 tells us that P must then be negative so long as neither P nor Q is 0. So, the A allele will sweep to fixation, eliminating the dominant A allele. A more interesting situation arises when the heterozygote is superior in fitness to both of the homozygotes. That is, W12 is greater than W11 and W12 is greater than W22, a phenomena known as heterosis. Hence, in this type of situation, for both alleles that are present in a population, equilibrium should be reached. Equation 2 confirms this intuition. It is easy to see that P is equal to 0 if either allele has gone to fixation. That is, if P is equal to 0 or Q is equal to 0, or thirdly, if the following condition obtains, where P into W11 minus W12 plus Q into W12 minus W22 is equal to 0. This reduces to P is equal to P steric is equal to W12 minus W22 by W12 minus W22 plus W12 minus W11. The steric symbol shows the equilibrium state, and this state is only satisfied when P is positive and there is presence of heterozygote superiority or heterozygote inferiority. It represents an equilibrium state of the population in which both alleles are present. This equilibrium is known as polymorphic, by contrast with the monomorphic equilibria that arise when either of the alleles has gone to fixation. The possibility of polymorphic equilibrium is quite significant and represents that natural selection will not always produce genetic homogeneity. In some cases, selection preserves the genetic variation found in a population. Such equilibrium is observed in natural populations as well in areas where the malaria and sickle cell anemia are prevalent. Individuals with normal genotype are eliminated because of malaria, while homozygous individuals for sickle cells are eliminated because of anemia. Heterozygotes, on the other hand, are favored individuals as they can counter the growth of malarial parasites because sickle shaped RBC does not allow the completion of life cycle, while one normal gene compensates for the oxygen carrying capacity. Numerous other aspects about natural selection can be studied by incorporating a parameter that measures the fitness differences between genotypes, such as rate of evolutionary change, which in turn has enabled us to answer questions such as how many generations are needed for selection to increase the frequency of dominant A allele from 0.1 to 0.9. Also, if the given deleterious allele is recessive rather than being dominant, then how much time it will take to remove it from the population. The one locus model is simple so as to be applied to real life populations. Selection is rarely the only evolutionary force in operation because genotypic fitness is unlikely to be persistent amongst generations, and hence the Mendelian segregation cannot be held exactly. Nevertheless, one locus model illustrates the essence of population genetic reasoning and the attendant clarification of the evolutionary process that it brings: migration. Migration is the fourth and final factor that can affect its genetic composition. both immigration as well as immigration contributes to genetic composition of a population. If immigrants are genetically different from the population they are entering, this will bring alteration in the genetic composition of the population. The evolutionary significance of migration comes from the point that various species are formed of diverse subpopulations, mainly quarantined from each other but connected by occasional migration, and therefore the gene flow stems from the migration among subpopulations, controlling the magnitude to which these may diverge from each other genetically. Consider a simple model for analyzing migration that assumes a given population receives a number of migrants each generation but sends out no emigrants. Suppose the frequency of dominant A allele in the resident population is P and the frequency of dominant A allele among the migrants arriving in the population is Pm. The proportion of migrants coming into the population each generation is M. So, post migration the frequency of dominant A allele in the population is P dash is equal to 1 minus M into P plus MPM. The change in gene frequency across generation is therefore P is equal to P dash minus P is equal to minus M into P minus Pm. Therefore, migration will increase the frequency of dominant A allele if Pm is greater than P, decrease its frequency if P is greater than Pm, and leave its frequency unchanged if P is equal to Pm. It is then a straightforward matter to derive an equation giving the gene frequency in generation T as a function of its initial frequency and the rate of migration. The equation is Pt is equal to Pm plus Po minus Pm into 1 minus Mt, where Po is the initial frequency of dominant A allele in the population, that is before any migration has taken place. With increase in value of t, the expression 1 minus mt tends towards 0, making it evident that equilibrium is reached when Pt is equal to Pm, that is when the gene frequency of the migrants equals the gene frequency of the resident population. On the other hand, single parameter that affects the frequency of genes at a locus is the migration, but this is unlikely to be the case. So, it is necessary to consider how migration will interact with selection, drift and mutation. Similar to the mutation selection balance, the deleterious allele can be maintained in a population by having a balance between selection and migration. Also, the drift can cause the subpopulations of a species to genetically diverge. Migration opposes this trend. It acts as a homogenizing force that tends to make subpopulations alike. Even a fairly small rate of migration will be sufficient to prevent the subpopulations of a species from diverging genetically. This theory has been employed by theorists to debate on the evolutionary importance of group selection and also based on the factor that differences among genes in groups which are essential for group selection to operate are unlikely to persist in the face of migration. Let us now summarize what we have learned in this module. Hardy-Weinberg equilibrium is a simple mathematical illustration for distribution of alleles in a population. Hardy-Weinberg equilibrium is best described in panmictic populations. Several factors account for a large randomly mating population to remain at Hardy-Weinberg equilibrium, namely absence of migration, mutation and forces of natural selection. In its simplest form, the principle can be expressed mathematically as the relative frequencies of the dominant A and recessive A alleles in the population may be denoted P and Q, where P plus Q is equal to 1. The Hardy-Weinberg principle gives the relationship between the allelic and the genotypic frequencies. It states that in the offspring generation, the genotypic and allelic frequencies will be related by the following equation: F dominant AA is equal to P square, F heterozygous AA is equal to 2PQ, and F homozygous recessive A is equal to Q square, and P square plus 2PQ plus Q square is equal to 1. In nature, none of the factors operate individually, and all these factors are involved in a complex interplay leading to distribution of alleles in a population. Multiple subpopulations differentiate on this basis from a parent population and thus lead to evolution and speciation.